the inflammasome activations. And we try to understand the mode action of the creosol-mediated NOD3 inflammasome inhibition. So we test the creosol on the reactive oxygen species production in response to the LPS stimulation, because the ROS is an important regulator of NOD3 and pro-1 beta protein expression. So based on this data, we found the LPS treatment can induce a lot of ROS production, and the creosol can significantly reduce the ROS production in LPS active macrophage. And we also found that the creosol can also reduce ATP mediated ROS production, because ATP mediated ROS production is an important signal in the activation signal of NOD3 inflammasome. It controls the KAT1 activation. So then we found the creosol has anti-inflammatory activity by reducing the ROS production. Finally, we investigate the anti-inflammatory activity of creosol in a mice model. So we found the oral administration of the creosol and followed by the LPS injection into the mice, and we measure the cytokine level in the mice serum, and we found in the serum the IL-1 beta, IL-6, and the TNF-alpha level is reduced by the creosol. So and the creosol also reduce the NLRP3 and the COX-2, all is inflammatory protein expression level in the liver. We collect the liver tissue and measure the NLRP3 and COX-2 expression, and we found that the creosol can reduce these two kind of protein expression in the liver tissues. So in conclusion, we found the bamboo vinegar isolate from the bamboo cargo production, and we found one pure compound creosol in the bamboo vinegar. It can reduce the cytokine production, including the IL-6 nitric oxide, and also reduce the NLRP3, pro-1 beta, and reduce the NLRP3 inflammasome activation. And in the mechanism study, we found the reduce the ROS production and the PKC phosphorylation is the major pathway how the bamboo vinegar inhibit the inflammation response. So finally, in the future work, we would like to test the bamboo vinegar or the bamboo vinegar pure compound using the different disease-specific inflammatory model. In my lab, we established several disease-specific cell models to mimic the disease-induced inflammation. For example, we can use the fatty acid to induce inflammation in mimic the diabetes condition. And we also can use cholesterol crystal to mimic the atherosclerosis, and also the uric crystal to mimic the gouty inflammation. So we can use this cell model to investigate whether the bamboo vinegar can be used to prevent or to treat this specific disease condition. And after the cell model, we also can use the corresponding mice disease model to test the bamboo vinegar in the mice disease model. So we can use a renal disease model, and we have some metabolic disease model. We also have some acute inflammation model. For example, the sepsis model or the inflammatory bowel disease model. So in the future, we want to continue my study about the bamboo vinegar to identify the mechanism or identify the compound can be used to prevent the inflammation. Okay, that's some of my preliminary data about the bamboo vinegar. So thank you very much. Yeah.
Thank you. Thank you for the Dr. Hua. And I thank you for you to open the secret for the bamboo vinegars, because we all know the bamboo vinegar is really good for the biotech. But I think from your presentations, we really find out how it works. And I can see the future is a lot of the good opportunities for the industries to using bamboo vinegars, try make different type of medicines for different disease. So this will be the one of the first time for our bamboo tech forums. We try to really going to see how the bamboo vinegar work. And I'm glad you can show the some of the scientific evidence to see bamboo vinegar is really useful. And just one quick questions. Your VV4, how is the temperature for the VV4?
Yeah, it's around 145 to 150 degrees. Yeah, it's a higher temperature.
So higher temperatures vinegar will much better than in use in the medical industry.
Based on the data, it's a little bit potent higher temperature. Yeah.
Okay. Thank you. Thank you for the speech.